Hello everyone, I'm Gregory Schulz, a professor emeritus at the University of Florida. It's my pleasure today to talk to you about battling biofilms and winning the war in wounds. I have listed my disclosures and conflict of interest, but all of my information will be based on data and I will not endorse any products. The objectives for my presentation today are to review the contribution of bacterial biofilms to chronic infections in 18 other clinical pathologies, and then to understand the link between chronic inflammation caused by both planktonic and biofilm bacteria, leading to elevated protease activities that destroy proteins that are essential for healing, such as extracellular matrix proteins, growth factors, and receptors. Also, I want us to understand and recognize the high tolerance of biofilm bacteria to most antibiotics, antiseptics, and disinfectants. And finally, I want us to describe the key principles of biofilm-based wound care that emphasize debriding of biofilms and preventing the reformation of biofilms as part of the step down then step up approach for effective therapies in chronic wounds with biofilms. As shown in this diagram from the excellent paper review by DePozo and Patel in 2007, they listed 18 clinical pathologies that are due to chronic infections caused by medical biofilms. Now, these include things such as chronic sinusitis, orthopedic joint infection, urinary catheter infections. But notice in 2007, there wasn't a specific pathology related to chronic skin wounds, and that's because the field really hadn't begun to appreciate the critical role that bacterial biofilms play in promoting chronic wounds and preventing them from healing. Now, the presence of biofilms in wounds doesn't prove that the biofilms are actually making a significant contribution to the failure of wounds to heal. And in fact, it could just be coincidental. So data, particularly from Tom Musto's lab, shows that when mice were created with full thickness skin wounds on the backs and then inoculated with Staph aureus or Staph epidermidis, and the healing rate measured, you can see there's a significant reduction in the rate of wound healing. But that still doesn't prove it's the biofilm component that's doing it. So the key when they repeated the experiment was to simultaneously treat the wounds with the inoculating bacteria, but also with a specific quorum inhibitor, the so-called RIP peptide. And that specifically and only blocks the production of the biofilm. When that happened, there was no reduction in the rate of healing because the mouse's superb immune system could control, engulf, and kill the planktonic bacteria, which prevented the biofilm from forming and was able to reduce the chronic inflammation that delayed healing in the non-biofilm inhibited mice. Now, biofilms are present in at least 80% of biopsies from chronic wounds, but only in a small percent of acute wounds, as reviewed in the meta-analysis by Matt Malone. And in panel B in the middle, you can see a typical scanning electron micrograph of a mature biofilm on the surface of a wound bed. But equally important, as I'll show you, is for us to understand that in addition to being on the surface, biofilms can also form under the surface of the wound bed, as shown in panel A, and some of the components of the exopolymeric substance from biofilms, which certainly contains polysaccharides, proteins, lipopolysaccharides, but also, as shown in panel E, the presence of free extracellular bacterial DNA, which is a significant stimulus for our innate immune system. Our toll-like receptor 9 actually recognizes unmethylated CpG sequences within only bacterial DNA. And so our immune system has evolved the ability to rapidly recognize when a bacterial biofilm is forming, generate the rapid immune response in an effort to try to prevent the biofilm from forming into a mature state. One of the key concepts for us to understand is how does the immune response to biofilms cause tissue damage and impair healing? And in this excellent paper by Bill Costerton in Science in 1999, as in panel A, he showed that in most cases when we have a contamination with planktonic bacteria, our antibodies, our antibiotics, and our immune cells are able to prevent the planktonic bacteria from multiplying to such a level that it will cause an infection and impair healing. But if some of those planktonic bacteria can attach, begin to quorum sense, form a biofilm structure shown in panel B and panel C, that begins to impair diffusion of our antibodies into the biofilm matrix, also our antibiotics, and our immune cells have difficulty in engulfing and killing this massively large biofilm community. And so as shown in panel D, what that results in eventually is a chronic inflammatory state where particularly neutrophils secrete large amounts of proteases and reactive oxygen molecules, which can lead to off-target damage and impaired healing. So why are these bacteria and biofilms so hard to kill? Well, there are multiple answers. The exopolymeric matrix can impair diffusion. It can can chemically react and neutralize many microbicides. It also is usually negatively charged, so it can bind and retard many positively charged cationic molecules like silver or antibiotics, PHMB. But probably one of the most important is, as I'll show you, that there can be persister bacteria in the biofilm that have low metabolic activity, and antibiotics only kill metabolically active bacteria. The metabolically active bacteria on the shell of the biofilm can consume oxygen, which can promote a niche region for anaerobic bacteria to grow, and most of the biofilms in chronic wounds are polymicrobial, so there can be a mixture of both genetic resistant bacteria that can provide bystander resistance proteins as well as enzymes that, such as catalase, that destroy hydrogen peroxide. To show you just real quickly how biofilms are so tolerant to antibiotics, if you see in the left panel where we grow planktonic Pseudomonas in broth and expose it to tobramycin, you can see that in 4 hours all of the bacterial biofilms have been killed, but in the biofilm phenotype, exposing those bacteria to 4 days of the tobramycin only reduces a log unit. And this is because, as shown in the right panel, many of the bacteria are not metabolically active in the center of the biofilms, and again, only metabolically active bacteria are destroyed and killed by antibiotics that inactivate and inhibit many of the bacterial essential proteins. So one of the key concepts for us to understand about healthcare is that debridement is the most important treatment for biofilms, but it usually does not remove all, and therefore it must be used in combination with other antimicrobial treatments that can kill biofilm or reduce planktonic biofilm from reforming biofilms. And another important concept is that slough, while it is an important component that needs to be removed from wounds, is mainly fibrin, and it is not all of the biofilm. In fact, when we look at the surface of a typical diabetic foot ulcer, we don't see a biofilm structure, but looking at the scanning electron micrographs from biopsies of that wounds, we can clearly see that there's mature biofilm present. And in addition, many of the bacteria and biofilms are under the surface of the wound bed, as shown by Thomas Bjornhardt's data in biopsies from chronic wounds. Staining for Staph aureus or Pseudomonas biofilms with selective reagents, you can see that they can penetrate 60 to 70 microns below the surface of the wound bed. Now, the biofilm is illustrated in Steve Percival's review, can be both on the surface and under the surface of the wound bed, as I've shown you, but they can also be present as a component in slough, but also in the wound dressings where they can shed microcolonies of biofilm back onto the wound bed, and therefore we need to use antimicrobial wound dressings in combination with the dressing debridement. This leads us then to this concept of step down then step up treatment for chronic wounds, in which initially The most aggressive and effective debridement is used to remove the biofilm. It's combined with antimicrobial dressings that will prevent the biofilms from reforming. The dressing and wound treatment can step down to maintenance debridement with more reasonable wound dressings, and if the wounds start to heal, fine. But if the wounds need additional stimulation to promote healing, then we can step up to advanced therapy such as growth factors, skin grafts, advanced cell and tissue products, and they will work wonderfully well because the wound bed has been prepared. So I hope this gives you an appreciation for the importance of biofilms in chronic wounds and ways to recognize and treat biofilms as part of your wound care strategy.